Hello everyone, and welcome to our online lecture series for Bio2. Today, we finally get to our organisms with membrane-bound organelles, the eukaryotes, collectively known by a number of different names that will likely change over your lifetime, as many of these phylogenetic groups have already changed both throughout my education and even in my time here teaching at West Shore. So that just goes to say that we know that all these groups are related, and we know that they're all eukaryotes, but we don't know exactly how they split up. It's very difficult to tell because we weren't there, and this happened a long time ago, and the evidence is incredibly mixed due to a lot of horizontal transfer. So, to go over our protist lineages, on account of the fact that in this course I do expect you to be able to draw out some of these lineages, let's make sure that we have that we are on the same page in terms of how we'll be drawing this phylogenetic tree. So, for now, in this class, we will consider there to be 6 distinct groups of protists. They all are eukaryotes, so they're branching down from our eukarya tree, which we know either is sister to all archaebacteria or is potentially even emerging from archaebacteria. Phylogenetics is complicated. But those 6 lineages are the archaeplastids, or the group that has the oldest ingestion of the endosymbiotic plastid, or the chloroplast. The chromoalveolates, rhizaria, and even nowadays sometimes they connect these two in an SAR group. More on that later. The excavates, these we know are fairly distinct and a divergent group from our protist polytomy. The amoebas, or the amoebozoans, and the opisthecons. And all of these groups are contained here within this image. So, not that you need to know any particular one of these by sight, but do note that there are a lot of different life forms, whether they be multicellular, or unicellular, or differ in their shapes, or hairs, or life patterns, or what they eat. The bottom line with protists is that they are incredibly diverse in lifestyle. It's kind of going out of fashion to consider this group monophyletic, but we for now will at least consider them off of the eukaryotic branch. More to be uncovered, I'm sure, for a very, very long time. They are defined by a polytomy. And this is in large part because the early members are unknown. They are frequently unicellular. With some of the largest cells in the world. Rather, some of the largest cells, like the genus Caulerpa, that can be up to a meter in size. That's one giant aquatic cell. Not only are they diverse in their lifestyle, but they functionally are able to achieve complexity without multicellularity. In many cases. So they've been able to subfunctionalize. They've been able to develop incredibly complex lifestyles without having to develop tissues. That's the bottom line there. No tissue specialization, but individual cell subfunctionalization. They can exist, so in terms of lifestyles here, they are able to exist sometimes as colonies, meaning cells that are loosely associated but not physically necessary to one another. They oftentimes have or can have many nuclei. And those nuclei can have different purposes. Like here in this paramecium, a great example of a protist, we see that they have a macronucleus and a micronucleus. The macronucleus is for general life function. The micronucleus is for reproduction. So it's pretty crazy that you can have different nuclei for different components of your life cycle. And this is exactly what I mean when I say complexity without multicellularity. In a single cell, they've developed organelles that are able to do things differently. Rather than develop a line of cells that's specialized on reproduction, like us, our gametes, they instead have an individual organelle for it, or can. They also are frequently defined by a protective layer on the outside. Some of them have a hard silica-based outside. The other very common outside is a pellicle protein outside. So the hard silica shell just basically adds an incredibly rigid outer structure, whereas the pellicle protein shell gives you some layers that provide flexibility. So there is a difference between these two, but they're, on account of the fact that they're oftentimes unicellular, they are very, very protective of that single cell. Their metabolism is incredibly variable. And honestly, as is the case with most things protist, the more time you spend reading about protist metabolism, the more new things you discover. It's almost a linear relationship because there's so much diversity here. The more time you spend, the more things you will discuss. So keep that in mind on account of the fact that we will discuss this very briefly, meaning that we're only covering a very small section of all of the possible protist metabolism forms. So they generally can be classified as either aerobic, or those that utilize oxygen, or anaerobic, those that do not utilize oxygen in their metabolisms. They can be mixotrophs, meaning that they both are using light to eat and food to eat, meaning they're both somewhat photosynthetic and somewhat heterotrophic. Or they can be frequently heterotrophic. And depending on the lineage that we're talking about, you may see that some of these evolve as originally heterotrophic, but now are more autotrophic. So it's absolutely nuts that you can get these massive changes throughout the life history. The heterotrophs have a couple of unique organellar structures. The heterotrophs can have a food vacuole, whereas plants will generally have a water vacuole to hold on to water. These can have a food vacuole or a special feeding structure. Like the mouth pore here on our paramecium. They have what is called a phagolysosome. This is a combination of the food vacuole plus an acidic lysosome, meaning one of these organellar structures that actually goes in and adds acid to the food. So it's kind of like a as you need it stomach. We engulf the food in a food vacuole, bring it inside via endocytosis, and then break it down by adding acid to it. It's very akin to the way your stomach works, but without having a stomach always present. So it's kind of like you make a stomach when you need it. Again, very unique. They will get rid of their food by exocytosis, which is kind of the equivalent of pooping. They get rid of any waste, and they even sometimes have a region of the cell that's doing this, like in the paramecium here, the anal pore. Weird to think about you forming an anal pore or an anus only when you need it. They're very, very flexible because of that unicellular lifestyle. They have very particular needs in terms of motility. Oops, forgot a letter there. In terms of motility, you will see cilia, or tiny hairs, that allow like beating back and forth to move things around, like embodied in the paramecium. Not only does the paramecium use cilia to move around, but it also uses it to push food into its mouth pore. In amoebas, you have pseudopods, or false legs. Just like you have a stomach on demand or a anus on demand, you can have legs on demand in some of these organisms. Pseudopods will be extensions of the plasma membrane and cytoplasm in order to push the animal forward, and then or the protist forward, and then roll towards that direction. And then the last major mode of motility is a flagellum, a long tail-like structure that we're very familiar with, and it's oftentimes indicative of what group these protists belong to based on how many flagella they have, and even at what angle their flagella grow. Weird, right? In terms of their ecology, just like their lifestyles, they're incredibly diverse, but they are oftentimes food sources for larger multicellular organisms, but they can also be animal and plant pathogens. Like Phytophthora, one of the most studied plant pathogens. It was responsible for the potato blight in Ireland, so the Irish potato famine that caused the large wave of Irish migrants, of which one of which was one of the descendants of my wife. So Phytophthora definitely had a direct impact, not only on my wife's family, but subsequently on my own, because, hey, I met her because her descendants came out this way. And then, so that is Phytophthora infestans. Oops. It's an oomycete. And then they oftentimes are defined, I'll squeeze this last point here, is that their life cycle has a protective stage that we call a cyst. So it can remain as a cyst in the environment up until conditions are nice, and then it will begin its procreation from there. So very diverse. They get their resources in a lot of different ways. They are a polytomy, so we're not quite sure as to how exactly they are related to one another. But we do know that they are related to one another because they share these fundamentally critical 8 characteristics. So now we'll go over the shared 8 characteristics. Because you might be asking yourself, if these are so diverse, how on earth do we know that they are actually a group? How come we don't assume that they are different from one another and therefore separate lineages? Well, these shared characteristics are both incredibly difficult to have evolved multiple times and with such similarity in their current functionality. So we assume that whatever shared ancestor of the eukaryotes there was, that it contained a lot of these features, and from there began to diversify. The first and most important shared characteristic is the presence of a nucleus and a nuclear envelope with pores. As far as we've been able to tell in all kinds of electron microscopy of different protists, we always see that their nucleus is similarly structured with pores. So for some reason, that seems to be an absolutely critical feature of our protists. The next is the mitochondria, an organelle that we're going to spend a long time talking about today. But all of these protists contain mitochondria. Their mitochondria are similar in their 16S or 16S ribosome sequence, or certain regions of their mitochondrial genomes, suggesting that this is from a single engulfing event. We will talk about the non-singular engulfing events of the chloroplast, but we do know that the mitochondria entered this branch once. So all 8 of these would technically be tick marks on this branch here. 1, 2, 3, 4, 5, 6, 7, 8. Lots of little tick marks for us here. More on the mitochondria in a second. The third one has been kind of connected to the previous group with the archaebacteria, but we do know that a fully structured cytoskeleton with the identical actin, tubulin, and filament proteins are shared across all protists. Now, sometimes those protists use them to make pseudopods. Sometimes those protists use them to make cilia or flagella on the outside. But ultimately, they are all using them inside for similar features to move things inside of the cell. This internal movement is absolutely critical, and the formation and breakdown of the cytoskeleton, which we know so much about, is actually one of the things my wife did her PhD on. So I got to learn a lot about that through her work, is very similar across all these protists, and we are more and more uncovering how this cytoskeleton is also potentially shared with some archaebacteria. The fourth feature is the presence of the flagella and oftentimes cilia. Seems to be something that they all innovate with. Our fifth shared feature is the presence of linear chromosomes with histones. That is a couple of terribly written letters. Let's try that again. Histones. This organizational feature of using histones in order to keep your DNA from getting too tangled is incredibly complex and incredibly shared across all of our eukaryotes. Therefore, we assume that this is something that did not evolve multiple times, especially since a lot of those histone proteins have incredible similarity. You know, in my experience in plant research and studying plant centromeres, we can even take histone proteins from different plant species, so something like corn and rice, and they won't be able to differentiate. They can actually work perfectly fine in one another. They substitute 100%. And so a lot of these histones, while they may not be able to do that same substitutability, we can do really nice alignments to see that they have a lot of shared similarity in their structure. The sixth is that a lot of them are able to reproduce in some sort of mitotic fashion. So they do share this history of mitosis as a way of copying themselves, whether they're in a multicellular body and able to proliferate cells in tissues like that, or whether they are unicellular and can reproduce via some sort of budding. The seventh is that they, in lineages that are sexually reproducing, they oftentimes will sexually reproduce with meiosis. And this is best embodied by the meiosis that we see present in animal, plant, and fungal lineages, even though they are very disparately related, with plants being more distant than fungi and animals are to one another. Each of these groups will reproduce via meiosis. And then last but not least, our eighth shared characteristic is the presence of a cell wall, a protective layer on the outside. But you might ask, you don't have a cell wall, so how come this is an ancestral characteristic? Well, animals actually carry a derived form of this, where we've lost the cell wall. And the cell wall is actually lost in a lot of lineages. This is oftentimes the case due to something called related to Dollo's law, where it's things are not re-evolved, but rather it is easier to lose this characteristic, to lose the cell wall, than it is to re-evolve it. So you have this interesting situation where many lineages have actually lost the cell wall in many different occasions, but also that many different lineages have retained the cell wall and done slightly different things with it. So these are our fundamental 8 shared characteristics for our eukaryotic protist lineage. Even if they're a polytomy, even if we don't understand exactly how they're related, even if we may not consider them monophyletic in the very near future, we do know that these characteristics are shared amongst them. And whichever way it shakes out, these traits will make for excellent tick marks on that eukaryotic protist lineage. Now, we need to spend some extra time talking about the most important, or perhaps the most important driver of this that we know the most about. We don't know exactly how a nucleus forms. We're uncovering more about the cytoskeletal history by looking at archaea. Flagella and cilia are so basal that they seem to be, even shared with some of the bacteria. The chromosomes with histones, we don't have a good way of studying that just quite yet. But we do know a lot about this mitochondrial history through something called the endosymbiont theory. And again, just like the theory of evolution, this is a theory in and only that it is the absolute best guess that we have a lot of fantastic evidence for. So theory does not mean we second guess ourselves in science. Theory means, well, I guess we do second guess ourselves, but ever so slightly. We are open to contradictory evidence being presented, but as of yet, we haven't found any. So keep looking, but I do think that the endosymbiont theory is incredibly compelling. The endosymbiont theory was first proposed by Dr. Lynn Margulis, whose name will crop up a couple of times because she had some amazingly innovative ideas and observations as to how the mitochondria could have come about. And I believe that she is currently working on a very relevant question to our research here, whether the nucleus or the mitochondria came first. So I'll be interested to see what her thoughts and conclusions and evidence are as to that end. But in regards to her endosymbiont theory proposal, she first observed that the mitochondria shares a lot of its genetic sequence as well as a lot of its features with alpha proteobacteria. Again, this is one of those many different types of proteobacteria that are incredibly diverse. And as an example of how diverse alpha proteobacteria can be, those of you who have taken Bio 1 will be familiar with Agrobacterium tumefaciens. That is the gall-forming bacteria that we use in plants to perform genetic modification. This is actually, alpha proteobacteria is or Agrobacterium tumefaciens is an alpha proteobacteria. So they can range from things that were our endosymbiotic mitochondria, as well as things that are able to shove their genomes inside of something else and replicate within them and cause cancer in plants. So quite a wide range of functionality. So what traits do we see shared between the mitochondria and our alpha proteobacteria? What indications do we see that they actually have a shared heritage? Well, first and foremost, the mitochondria, just like our alpha proteobacteria, have a circular genome. Contrast that to our histone-wrapped linear chromosomes that we have within our nucleus as eukaryotes. Completely different, but really good evidence that there is shared ancestry. The second is the ribosome size. Eukaryotes, all eukaryotes, have ribosomes that are bigger than prokaryotes. Eukaryotes are known for having 80S ribosome size, whereas all of our prokaryotes have a 70S ribosome size. So that's true in both bacteria and archaebacteria, and for whatever evolutionary reason, whether it be purposeful or drifty, random, stochastic, eukaryotic ribosomes are bigger at 80S. Our mitochondria, however, have a 70S ribosome. So, ignoring the fact that you already have tons and tons of bacteria living inside of you, you also have these alpha proteobacteria endosymbionts that still carry the ribosome size of 70S inside every single one of your cells. So, the ribosome size, I think, is pretty good evidence of this shared ancestry. That the fact that mitochondria use this very unique and different ribosome size suggests that we may be two different organisms occupying the same house peacefully. The third and very interesting and weird observation is that some mitochondrial genes have migrated to the eukaryotic nucleus. Now you might ask why this has occurred. Well, first off, you can go ahead and assume that when you have this kind of migration, that there is two different organisms living in the same place, but it is fairly weird to see genes migrate from one to the other. And not the opposite way. They're not going from our circular genomes in the from our linear genomes in the nucleus to the circular genomes in the mitochondria. They're just going from the circular genomes in the mitochondria to the nucleus. And this is because it allows the nucleus to control some of the reproductive capacity of our mitochondria. They are able to reproduce themselves. They reproduce themselves in a process called binary fission, which will be the sixth fact down here, this sixth shared feature. And by controlling some of the genes that would normally be found in the mitochondria, the nucleus of a host can actually control when and how frequently the mitochondria is able to reproduce. So that is pretty good evidence that there is this shared history. The fourth is that when we try to take the mitochondria out of a eukaryote and have it live on its own, it does not. The mitochondria actually needs its host. So unlike its alpha proteobacteria ancestor, the mitochondria has now developed a dependency on the eukaryotic host as evidence of its endosymbiont past. The fifth feature is what I consider to be kind of one of the most nail in the coffin. Like if I were Lynn Margulis and studying this topic some time ago, when I got this bit of evidence, it really would have spurred things along to make me realize that mitochondria may have been these endosymbionts. It is the observation that they have double membranes. Whereas your Golgi body is a single membrane and your nuclear envelope's a single membrane, your mitochondria is double membrane bound. But those membranes are not the same. The outer membrane has proteins that are specifically eukaryotic, whereas the inner membrane that would have been the plasma membrane of the alpha proteobacteria has proteins that are specifically prokaryotic. So even the protein structures that are present on these different membranes show this really, really ancient blueprint of previous life as an alpha proteobacteria. Additionally, the cristae folds inside of the mitochondria are carrying prokaryotic specific proteins. So you have all of this evidence: circular genomes, just like alpha proteobacteria; ribosomes, just like alpha proteobacteria; genes that have migrated to the nucleus, showing a dependency; the fact that they need a host to survive, showing a dependency; showing a past of a previous engulfing event with that double membrane bound organelle that's different membranes on the outside and inside; and lastly, reproduction like alpha proteobacteria gives us really solid evidence and confidence in the endosymbiont theory. And so here we have a diagram from your book that illustrates how it may have happened. Originally, we have had a proto-eukaryote, larger cell, not quite yet membrane-bound organelles, potentially DNA organized maybe in with histones, either circular or linear. But then the infolds of the plasma membrane of an ancestor prokaryote give rise to the endomembrane components. From those infoldings, you get the nucleus and the endoplasmic reticulum, and eventually, potentially, the Golgi body. From there, the first endosymbiotic event occurs, where an aerobic bacteria, the alpha proteobacteria, gets put in and evolves into the mitochondria. In a second endosymbiotic event, you actually get this evolution in chloroplasts. So we'll talk about chloroplasts as a second endosymbiotic event. But our first basal lineage of all of our protists would have had this mitochondria, and the mitochondria shared both with plants as well as all of the other protist lineages. So hopefully that kind of illustrates how this would have happened and how this feature would be shared across so many different protists. Let's take a second now to talk about a parallel event occurring later within a very specific lineage, the archaeplastids, and how it gave rise to the chloroplasts. The chloroplast is, of course, the organelle responsible for photosynthesis, the process by which plants take in carbon dioxide and water and convert it into sugar and oxygen, the oxygen you and I breathe. In their own independent endosymbiotic event, as illustrated in arrow 3, we had our heterotrophic eukaryotic ancestor that then engulfed a cyanobacterium in a primary endosymbiotic event. One of these membranes surrounding the plastid are lost, and so there's a little bit more flexibility in what exactly is happening with the membranes of the chloroplasts, so I won't focus too much about that. But we know that some of the membranes can be lost rather than retained all of the time. Cyanobacterium seem to have their own kind of double membrane thing going on with the thylakoids inside. After this endosymbiont is fully engulfed and incorporated into our archaeplastid, we then got a secondary endosymbiosis. So from this lineage, this group that branches off would have gone on to make our plants, a single endosymbiotic event. But this ancestral group, before they evolved multicellularity in the plant lineage, one of them may have been engulfed, and likely were engulfed, in another protist lineage that then has more membranes and a vestigial nucleus inside of the plastid. So sometimes you will get chloroplasts that have 2 membranes, and sometimes you can get them with 3 or 4 membranes. So multiple membrane bound, if indeed this secondary endosymbiotic event is true. So, you can imagine if a protist can engulf something like a cyanobacteria once, perhaps a bigger protist can engulf something bigger a second time. And if that bigger thing happened to also have eaten a cyanobacteria, now all of a sudden you've got a double-wrapped chloroplast. So this plastid endosymbiosis has its own evidence and history of storytelling. The perhaps best line of evidence here, or a line of shared ancestry, the biggest thing that we're looking for, is the presence of chlorophyll A, which is a very specific type of chlorophyll pigment that is shared across all of the different plastids, as well as some of the other accessory pigments. The fact that this is shared across all the archaeplastids and any of the other lineages that have similar plastid-like organelles is pretty good evidence. We have a unique feature in that it's multiple endosymbioses, multiple engulfing events. The oldest is that of a cyanobacteria, but has lots of descendants, whereas the new seem to be in a couple of different lineages. So we have the old in archaeplastids, and this was the engulfing of a cyanobacteria, whereas there have been several newer events occurring within amoeboids, rhizarians, and Pollinella being the absolute newest, so a single lineage that seems to have a new endosymbiotic event. The fact that the cyanobacteria has thylakoids and that we in our plants, I say we as though I am a plant, sorry, our modern day plants have thylakoids structured similarly, is good evidence. So other evidence that we have, again, circular DNA. Let's see if we can write a little neater. Circular DNA rather than linear DNA, showing an endosymbiotic history. The double membranes, but also triple and quadruple membranes. Ribosome size is again shared, where the chloroplasts have a 70S and their eukaryotic hosts have an 80S size ribosome. We also see the gene transfer, so genes in nucleus, just like we saw within the mitochondria. The sixth line of evidence, or fifth, skipping 5 here, fifth line of evidence is actually within that Polynella lineage, where because this is such a new endosymbiotic event, the chloroplast here still has a peptidoglycan cell wall. And that's pretty nutty. The fact that it's so recent that the cell wall in the chloroplast, like organelle, has not gone away. And then, again, the secondary endosymbiosis is really good evidence that this has occurred, because if it occurred once, we see it happening multiple times. And so here I actually found this image on Wikipedia. I think it's actually quite clever to show the relationships between some of our major domains, the domain bacteria, the domain archaea. And I wanted to have a quick discussion with y'all as to why some of these are connected. So we know that, or what is the identifying feature that will allow the artist here to connect these different groups? Why does archaea contribute to the protists or protista? Well, here we know that it's the cytoskeleton, the cytoskeletal features. How about why does bacteria, or which lineage of bacteria contributes to protista? Rather than descent, this is one of those horizontal transfers as a mitochondria. So this branch here shows the addition of a particular lineage of bacteria in an engulfing endosymbiotic event as mitochondria. What about this lineage here? Why do bacteria contribute directly to the lineage that contributes to all plants? Well, this is our endosymbiotic event for a chloroplast. And then you got to go, what's going on here with our red plant leaf that contributes to a brown lineage of protists that kind of branch around? Well, this is our secondary endosymbiotic event of an archaeplastid in which some lineage of protists receive the plastid from shared ancestry with some of these early diverging archaeplastids, particularly the red algae group. So we'll talk about that in a second. But you can see how all of these branches are not completely separated on account of the fact that they can have these horizontal transfers of either engulfing entire things or, on a smaller scale, not something that's discussed in the tree, but individual genes popping from one to the other. You can see that animals and fungi are directly descended from protists, but and are more similarly related to one another than they are to plants, even though multicellularity has evolved here, 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 as our most recognizable forms of multicellularity. So I thought that was interesting, as it was an illustration of discussing basically the things that we were going into greater detail here with our endosymbiotic histories. So now we transition the lecture to discussing some of the specific characteristics of these different lineages. So now it's like a rapid-fire National Geographic through our eukaryotic supergroups. A little bit more resolution here with some more connected points as an image from your textbook, but we still have our 6 major lineages, the archaeplastids, the amoebozoans, and opisthecons. We'll note that they are more similarly related to one another, so we oftentimes will draw them as connecting to the same point. Your book does that in a slightly different way. The rhizarians and chromoalveolates, though more recent research is again suggesting that these two might be more closely related to one another, with the rhizarians, alveolates, and stramentopiles all kind of forming one polytomy. And then the last but not least, we have the excavates, which we know are a separate lineage. So for purposes of this class, and because these are still sometimes hypotheses that are being developed, I'd like y'all to keep track of the tree like this, with 6 separate lineages, amoebas and opisthecons more closely related to one another than they are other lineages. We know archaeplastids are their own separate lineage, and the others are a very, very unresolved polytomy that may change over your lifetime. So let's go over the different components of each of these groups. So starting with the red algae, we see that this group is the most basal of our archaeplastids, that they have a pigment called misspelling it, erythrin, that gives them their own color. The reason for having this different color is that it is actually a deeper water pigment, so it allows for the absorption of light at a deeper depth, so that's kind of a unique feature of red algae. After red algae, we get the diversification of the chlorophytes, a basal green algal lineage that includes organisms like Chlamydomonas, which we'll talk about more when we get to our plant discussions. Caulerpa, that absolutely huge celled group, and Volvox, a really cool lineage of circular algae that are real neat to look at under the microscope. We then have the lineage of green algae that are sister to all land plants. These are the charophytes, and included within the charophytes is Chara. Moving on from our archaeplastids, we have the amoebozoans, which are defined largely by the presence of pseudopods. They have frequently lots of nuclei. So many of the amoebas have lots and lots of nuclei. And they also share this weird feature where they have a loss of the mitochondria, which is quite interesting. The opisthecons are a lineage that are defined by having a single posterior flagella, so a tail, as it were. And within the opisthecons, you get both your multicellular animals and your multicellular fungi. These different lineages are both characterized by being heterotrophs. So heterotrophs with the tail end that eat other things. Now, animals have evolved in order to eat other things inside of them, whereas fungi do their digestion on the outside and then absorb the nutrients after things are digested. These are saprotrophs, eating things on the outside by decaying them. Next up, we've got rhizaria, which are defined by having thin pseudopods for feeding. those pseudopods are really enabled by the presence of a cytoskeleton and similar cytoskeletal features. And other rhizarian traits that occur commonly are calcium carbonate and silica shells. So these are quite beautiful organisms that are oftentimes carbon sinks in our aquatic environments. They will absorb carbon from the air and sequester it, and then die on top of the ocean, sink to the bottom, and bring that carbon down with them. They do have these very, very cool shells. The chromioalveolates, and again, this group can sometimes be, or is now being referred to as the SAR group, the Stramenopial alveolate rhizarian group. The chromioalveolates are really, really cool and do show that secondary endosymbiont event where one of the lineages of the chromioalveolates, one of their ancestors, engulfed a red algae, and now they share more of the chlorophyll pigments in common with red algae than they do the green algae. But we've also seen that this secondary red algal endosymbiotic event has also been lost in some lineages. So it's gained and it's lost, and they've diversified, and what is going on? Because of this secondary endosymbiotic event, many of them are photosynthetic. And if they are not photosynthetic, they can oftentimes be pathogenic or causing disease. And here in the chromioalveolates is where you will find that paramecium. And I know since some of y'all have looked at paramecium under a microscope in a previous biology class, I'd like to give you guys at least a bit of a line as to where they're found. Paramecium are ciliates. The dinoflagellates are one of the many groups that we refer to as our oceanic plankton. But they are our plankton with a very odd shape. It's the best way to put it. They have a very, very weird feature in that they do not have histones. So on the dinoflagellate lineage, you can see histone loss as one of these shared characteristics. You might also recognize dinoflagellates as being the red tide, the bioluminescent organisms that cause very cool looking waves in the ocean. The apicomplexans, very long names are all of these, have a secondary endosymbiosis of the chloroplast. But also within this group, you find malaria. So, photosynthetic, but also malaria. Again, protists are very, very, very diverse. Down our Stramenopile lineage, the whole reason they're called Stramenopiles is because they have stiff hairs on the flagella. In some cases, they have stiff hairs on, or in many cases, they have stiff hairs on one flagella and then one unhaired flagella, so weird. Diatoms are also one of the major carbon sinks. You'll notice that a lot of these organisms are actually aquatic, again reflecting that ancient history of having evolved in water, so we'll see a lot more diversity of these protists in water than we do on land, simply because they've been there for longer. Diatoms also have a very cool silica shell. I encourage you to give them a quick Google to take a look at the beautiful patterns that are there. Brown and golden algae are, again, where we see this secondary endosymbiosis from red algae and sharing of that feature. And then we have the oomycetes, the water molds. And this group has popped around the tree of life. It used to be in different places when I was learning it, but currently they're grouped within the oomycetes. The best known oomycete is Phytophthora infestans. That lineage that causes potato blight and a lot of other blights. The excavates are incredibly unique compared to other groups because they are asymmetrical. And as it turns out, asymmetrical things are not very common on planet Earth. So we see much more commonly symmetrical organisms, as that seems to be what genetics can easier code for. But the excavates buck that trend. They're asymmetrical and always single-celled. Within the excavates, you'll find things like Giardia, the digestive tract disease that you do not want to get, as well as African sleeping sickness, a parasite that causes lethargy, as well as some other unfortunate side effects. So there's our diversity. There's our tree of life. We did a little bit of a sprint through some of the different traits of these different organisms that you can see along the protist tree of life. We know that they do share a common ancestor. We know of their 8 different features that are common across all of them. I encourage you to take some time to do some Googling. Oftentimes rely on Wikipedia for images of these different lineages. We'll end the lecture by going over some images of and seeing if you can guess what the major lineage that we're looking at is for these several images. Though again, if you go to Wikipedia, you'll probably recognize some of these. It's just a little bit of time to become familiar with some of the different lineages. Googling them to read about them on Wikipedia, to figure out some major features, something that sticks with you, a unique feature. I won't be asking you to memorize all of these different separate groups. Focus instead on the 6 supergroups: the archaeplastids, amoebozoans, opisthecons, rhizarians, chromoalveolates, and excavates. But it might help solidify that memory if you do additionally look up some examples. So here, what supergroup might we be looking at with this image? Well, it's a multiple lineage within the archaeplastids. We have the land plants, and then we have algae within the water. The green algae that occupies many of our waterways. So here you have the archaeplastids. This group here, any guesses? This is our SAR supergroup. Wikipedia has them upgraded, not just as the rhizarians next to the chromoalveolates, but instead lumping all 3 of them together into this SAR group. You can additionally find a link on Wikipedia to a TSAR group, the singular R group, so that way you spell Tsar like you would in a Russian, the once upon a time Russian lineage, which again, Tsar descends from the word Kaisar, or Caesar. There's a linguistics lesson for you, middle of a biology discussion. But you have a wide variety of different protist lineages in the SAR group. Next up, we have this kind of pixelated image of Giardia, which is an excavate. You'll notice the asymmetry here, lots of different flagella pointing off from different directions. How about this beauty? We have the amoeba with its pseudopods for moving. And then last but not least, we have a diatom, which, looking back up on your tree, always referencing the tree, you'll see that your diatoms are part of your chromoalveolates. I included a particular image of the diatom just to show you how cool that shell is on the outside, and encourage you to maybe do some further Googling for some cool patterns on different diatom shells. Notice the symmetry here compared to the asymmetry visualized in the Giardia. So that'll do it for our exploration of our protists. We've now established the base of our tree of life with our 3 domains, the bacteria, the archaebacteria, and eukarya, and looked at the polytomy that describes all of our protist lineages. From here, we'll start venturing down some of our more multicellular groups. We will be exploring the archaeplastids and the opisthecons for the remainder of our biodiversity discussions. Yeah, that means there's a lot of diversity down those 2 groups. See you next time when we start discussing the multicellular things on the tree of life.